Okay, I'm Denver Holt of the Owl Research Institute, and our crew is out here today on one of our research projects. This is the long-eared owl research project, and most of the people here are the crew, although this is our volunteer from Explore, Courtney.
Hi. That's
Steve Hyro from the Owl Institute, Matt Larson, and Liberty de Grand Prix. And this project here is now in its 30th year to study the long-eared owl. It's a long-term, year-round research project looking at the populations over time. We do a lot of other things, such as genetics and hormones and food habits and nest sites, but probably the most important thing is to be able to do the research throughout the year, do a marker recapture, and try to assess what the population is doing over time. In this area, our study area, we now know the population has been declining for some time now, although we're having a little bump this year, and we're having a good year. So we've captured these birds. We keep them in hand. We know they're stressed out, but we also know after we release them, they just go right back to business and get back into their roosts, and then we leave them alone until the next trapping session, which could be 1 to 2 weeks from now. So we're going to show you what we do as far as measurement data is concerned, and then we'll release the birds after we ban them and take the measurement data and move on to the next one. Okay. So now we know the birds are stressed out, so don't worry. This is just, part of wildlife research. And Matt is going to band the bird here. I'm going to hold it, and it's very standard stuff that we do. And so he's going to take this band. Okay. And he's going to give it its own little identification mark, or a little bracelet here. And this is all registered with the Bird Band Laboratory in Laurel, Maryland. All the numbers will be sent in, and they'll be set up in a database and actually open to the public for them to use. And now it has its own individual marker. And that way, if we capture the bird over time through different years, we can tell you a little bit more about them, whether they're showing fidelity to a site, fidelity to a mate, where they may have migrated to. And, unfortunately, sometimes, if they get killed, hit by a car, or some type of mortality, the band, if someone finds it, turns it in, will allow us to gather that information. So that was the band. Now the next thing Matt's going to do is he's just going to measure the chord, or the wing chord we call it. And so this is just the arc of a circle. Matt's going to take these are pretty standard measurements data for all of us who study birds.
283.
And we always repeat it. Matt says it. Liberty's recording. We'll repeat. That was the wing chord. Now he's going to measure the tail. And so, we'll talk a little bit more. The birds are definitely stressed out when we do the research, but what we think is important is to share, wildlife research with the public.
155.
And so we feel if we share with the public, then we have a conservation ally. Maybe helpful in voting, protect areas, maybe helpful in funding for research. And I think it's just important to keep the public involved in what we do.
The eyes kind of lemony.
Okay.
Lemon.
Generally the eyes are yellow or a lemon. We don't know what the lemon color is, but we definitely do see it and we record it. We'll be able to tease that apart at some point in time and look at age and sex in relation to eye color. But right now it's kind of uncommon to have a lemon colored eye. They're basically just yellow. We don't break it down into shades. And so these are the things we record. Now Matt's going to look at the wings and the feathers. He's going to try to age it and sex it for you. And each bird will be in hand, 10 to 15 minutes, and then they'll be released. And we try to release them in an area where they're not recaptured. For those of you who are concerned about stress, absolutely the birds are stressed. We know that now. Through our research, we've been able to collect blood, look at their corticosterone levels, and even though they're stressed out in hand, we know that they will return to the roost sites when we're done.
Here we'll look at age of this guy. So we're looking at molt, age of the feathers. We have the primaries here. There's 10 primaries and 12 or 13 secondaries here. And we're just looking for differences in color and wear. Newer feathers will be a lot darker than older feathers, and you kind of look across the flight feathers here, and they're all pretty much the same kind of shade of brown. None are distinctly darker than the rest. So we're going to say it's one generation of feathers, or these are all the same age, which means either it's a young bird that hatched sometime last May, or is an older bird who had a complete molt. And we can differentiate that by looking at the underwing. A lot of times in the older birds, you'll see like this kind of fluorescent kind of pink wash here on the underwing lining. And we're not exactly sure what that is, but it's associated with new feathers, and only the older birds and long-eared owls will have that kind of pink wash. I don't see it here. So this would be a bird that hatched, most likely May or around May last summer.
Yeah, so even though it's full grown, it's still quite a young bird, 6 months old, 7 months old, something like that, 5 months old. Okay, next.
Next, we'll predict sex. We do that by looking at the
chart.
The color.
This is kind of a cool thing here, you guys, that we developed years ago. We use a soil color chart, that soil research is used to score soil color. And we developed this some years ago and published a paper on it. And we've got 96 out of 99 correct where we knew the sexes after we predicted it. We caught them as breeding birds. And Matt's going to look at this and he's going to score the bird. We're so good at it now that we can do it just on our own, but we'll show you the method.
So here's the soil color chart. Use it to describe, soil color, and so if you can see it, you can maybe pick out a color that kind of reflects the overall kind of coloration here of the underwing coverts, these feathers right here. And what we find over the years is that males tend to score up in here, and females tend to score more over here. And so depending on where you picked it, you might somewhere in here, one of these lighter colors. So we'll say this is a male, so a hatchier male, a young male.
Okay, in his first year of life, young male. So hopefully it makes it through, and that's where the banding will come in. If the bird, migrates or stays here the winter, there's an opportunity in the future for us to recapture this bird and then have some information on, its age, its mating status, where it settles in, or perhaps it leaves and someone else captures it, which is uncommon, or it gets killed and they turn the band in again, which is uncommon. But at least we'll have a marked bird that we can gather some information. And by doing this for all the years we've done it, we can tell you a lot about their mating system and whether there's fidelity to a site, fidelity to a mate, do young return to the natal area. So we have a lot of great information from just the banding alone.
Want to just show
okay, now Steve's going to walk in. He's just going to show you the difference here in the coloration of the female versus the male. Okay, you hold that and I'll hold that. And then you can see that. Okay, and once again, again, I told you the birds are stressed out and they're going to be okay. But we think it's really important for the public to be able to know what we do as researchers and why we do these things. Okay, now we're going to weigh it. We'll just shut her down for a sec here. I should have told you that